Welcome to clickmeproject.com. Let me discuss about the process prostate cancer detection fusion of cytological texture features. Let me discuss about the process prostate cancer detection fusion of cytological and texture features. We propose a framework which detects the cancer using the cell images we obtained. The cell images were initially pre-processed to remove the noises from them. Noises refers to the unwanted pixels in the image. For pre-processing, we are using some filters in order to remove the noises. So here we are using median filter. So the median filter removes the noises in the tissue images. Then the images are segmented using object likelihood generalization. The segmentation refers to the choosing the required region of interest. The features were extracted from the segmented regions using GLCM and mean and standard deviation values. So here we are extracting the texture based features and the mean and the standard deviation features from the tissue images. We also train the tissue images based on the features extracted. The extracted features were then passed into the classifier. Here we are using support vector machine for classification. The classifier classifies the images into normal or abnormal using the training features, testing features on the true labels that we are providing to it. Thus, we are identifying whether the input tissue sample is affected or not affected. Let me see the flow of the process prostate cancer detection fusion of cytological texture features. Initially, we have some images in the database. We do training process initially. So, for all the images in the dataset, the region of features were chosen and the features were extracted for all the images in the dataset. And we also give a true label, that is which represents the given image is a normal or abnormal image. So, we are also giving that and this process is the training. So, here also we apply pre-processing. Then we go to the testing phase, that is we test the process with the image that we have given. So initially when the test image is taken, pre-processing is applied. So for pre-processing we are using median filter. It is often disabled to perform some kind of noise reduction in image or a signal. Median filter is a nonlinear digital filtering technique often used to remove noise. Such noise reduction is a typical pre-processing step to improve the results of the later processing. Median filtering is very widely used in digital image processing. Then region of interest was chosen from the input image using likelihood binarization. So it is done based on the threshold segmentation. So regions within the threshold were grouped as a single region, and the regions that are different from the threshold are grouped into another regions, and they were separated by applying some specific colors or by replacing the colors with the original pixels in the image. Then we are extracting the features from the region. So for that we are using GLCM feature and the standard deviation mean and values feature from the image. So while extracting the GLCM feature, the texture informations were retrieved, and the mean and standard deviation gives the specified needed gray level value changes, and which represents the texture features of. Then finally we are passing the test image feature and the train image features into the classifier. For here we are using SVM classifier, and in machine learning SVM supervised learning models with associated learning algorithms that analyzes the data and recognize the patterns used for classification and regression analysis. The basic SVM takes a set of input data and predicts for each given image which of two possible classes forms the input, making it a non-probabilistic binary linear classifier. Given a set of training images, each marked belonging to one of the two categories, and SVM training algorithm builds a model that assigns new examples into one category or the other. An SVM model is a representation of the examples as points in the space mapped so that the examples of separate categories are divided by the clear gap that is as wide as possible. Using that we are getting the classified result. Finally we are analyzing the performance of the classifier that we are using. So the obtained false negative, false positive, accuracy, sensitivity and specificity values, it shows that the performance is increased when comparing to the other algorithms used. Let me see about the execution of the process prostate cancer detection fusion of cytological texture features. If you want to run the code, Select the file main.m, which is located in the source code folder. Right click it and click on run file. You will get the guide window which is needed to run the code. This contains all the user interfaces which are required in order to run the code. So initially, first train the database by extracting the features for all the images in the which is present in the database. So first give input image. We give this input image and we do code processing. Then region of interest is selected in the input images. Then features are extracted for the input images. So here we are extracting GLCM feature and mean and standard deviation feature. These values were extracted, and then finally we do classification. So for classification, when we click classify, the classified result is displayed. So the input image is displayed as abnormal image. Here the positive value to negative value, accuracy, sensitivity and specificity values obtained were given. Finally, thus we are classifying the input tissue samples by extracting the feature values. Here we have extracted GLCM feature values and mean and standard deviation feature values, and also we are selecting the region of interest from the tissue samples, and finally we are classifying it using a SVM classifier. Thank you.